Hello, everyone, and welcome to another edition of Contagion. My name is John Parkinson, and I'm the senior editor. Joining us today is Gilead's Romas Gelazounas, who is Executive Director, Biology, and Dr. Devi Sengupta, Senior Director, Clinical Research, Virology. Thank you both for being able to join us today.
Thanks very much for having us, John.
Thank you, John.
Absolutely. Let's jump right into the topic. Can you offer some background on Vesatolimod and its mechanism of action?
Yeah, John, let me take that question. So I think what I'd like to do is maybe frame the problem for you so that we can understand together how TLR7 agonist Vesatolimod work. So in the case of HIV infection, as you probably know and your audience knows, it is treated with antiretroviral therapy. And it has to be taken for life. And the reason is because HIV establishes a latent or dormant reservoir in the immunological memory. So these particular T cells that constitute the immunological memory, that's where HIV lands and is able to stay there dormant, sometimes active, throughout the life of an individual. So the challenge is to eliminate safely that particular reservoir because it forms a reservoir. So over 10 years ago, when we started this program, we first asked ourselves, what would be a good way to awaken this reservoir? And we started with these TLR7 agonists that we had been working on for the treatment of hepatitis C back in the day. And we did discover that over many years of research that these TLR7 agonists are able to modestly awaken HIV from a latent state. And as a result, we decided to collaborate with various partners to go test this concept in vivo. Now, in our case, we're very lucky. We have a very good animal model of AIDS. We basically have this simian immunodeficiency virus, which is the cousin of HIV. It infects rhesus macaques. Basically, if left unchecked, the virus basically destroys CD4 T cells and the animals progress to AIDS. So very similar to the human condition. Now, the important thing is we can treat these monkeys with antiretroviral drugs, the same antiviral drugs that we use to treat people. And you can, again, stop replication in monkeys and also establish a comparable reservoir as we see in people. So we then decided with a consortium of colleagues, Janssen, the United States Army, the MHRP program, and our close collaborator, Dan Baruch, who's a professor at Beth Israel Deaconess Medical Center, we decided to do a study to test basically the combination of a comparable molecule to Vesatolimod, a TLR7 agonist made by Gilead, together with a therapeutic vaccine, a lot like the one that is being tested by Janssen in clinical trials in South Africa, for example. And so when we combine those two agents, Vesatolimod-like molecule with a therapeutic vaccine, we treated these monkeys, we discovered that the combination together led to a form of viral remission. That means that when we stopped antiviral therapy, some of the animals showed recrudescence of virus, the SIV virus in this case, but the immune system of some of those monkeys, about a third of them, was able to control that viral infection. So a state of remission, if you will, that is the immune system of those monkeys was educated to basically contain that virus. So this was very exciting. It was a very exciting discovery for us, suggesting that you could potentially, in an animal model of AIDS, lead to, with this combination of agents, to a remission. We then did a second study a few years later where we combined this TLR7 agonist, Vesatolimod, with an antibody that binds to the surface protein of HIV, or SIV in this case. And we basically combined those two agents, and in that case showed that we were able to basically deplete these reservoirs of latent virus, such that the monkeys appeared to have been cured. So this is very exciting. These developments are very exciting to us because it tells us that a TOL7 agonist with vaccination or a TOL7 agonist with an antibody in preclinical animal models could lead to basically remissions or potential cures in monkeys. So this is why we became very excited about Vesatolimod, the TLR7 agonist class in combination therapies, and we were really excited to translate these concepts to the clinic. And that's what Devi has been doing for the last several years and her team.
Okay, very good. For those not familiar with Gilead's research on this therapy, can you provide an overview of the study associated with it and what the findings were?
Sure, John. So the first thing I'll say about the study that was presented at the AIDS 2020 conference this week is that it was really a large team effort that spanned across several institutions. And the principal investigator for the study is Dr. Steve Deeks at UCSF, but many other institutions were involved as well. And most of all, we're extremely grateful to the study participants who committed countless hours and of their time to be part of this study. So as Romas mentioned, we believe that we now have a very strong foundation from our monkey work for our cure program at Gilead. And the first step in translating those monkey results is to establish the safety and the activity of Vesatolimod and other agents in people who are living with HIV. So in this first study that, again, was presented at AIDS 2020 this week and also at CROI earlier this year, we hypothesized that people living with HIV who have partial immunity to the virus that allows them to naturally control the virus to some degree might benefit from an immune modulator like Vesatolimod that would then allow them to have better control of the virus when they stop their antiretroviral therapy. So in this small study, we randomized people to placebo or Vesatolimod and dosed them over about 20 weeks and then stopped their antiretrovirals in an analytical treatment interruption. And what we found was that first and most importantly, the Vesatolimod was well tolerated in people living with HIV. And then what we observed was that the people who received Vesatolimod had a modest delay in the time to viral rebound when they stopped their antiretroviral drugs. We also found that Vesatolimod enhanced antiviral immunity and was associated with a decrease in that latent or dormant HIV reservoir that Romas mentioned, as measured by a novel assay called the intact proviral DNA assay.
What do you find to be the most exciting aspects of the study?
So I think this study really represents the first clinical evidence that Vesatolimod might play a role in our HIV cure strategies that we're pursuing. And it also affords us the opportunity to really dive deep into the data and understand more about the mechanisms of Vesatolimod and how we can exploit those to better achieve remission in people living with HIV.
Very good. And the next steps for this emerging therapy?
Well, we're really excited about our next steps, which are centered around combination therapies. So as Romas described, in the monkey model, we have seen that TLR7 agonist, when combined with a vaccine or a broadly neutralizing antibody, can induce cures or remission. And so we've now moved to that next step in the clinic. And we have a number of trials that are either ongoing or planned in the near future, including one that we're really looking forward to the results of, which is a collaboration with a biotech company called Alix based in Spain. Alix is developing a therapeutic HIV vaccine, and we are combining that with the Vesatolimod in a study of people who started ART early in their infection.
Excellent.
And we also have other trials planned with broadly neutralizing antibodies as well. So we're looking forward to those results over the next few years.
Excellent. You'll have to keep us up to date on everything as things proceed.
Absolutely.